persons commit suicide. Why? Because they are so severely depressed and they feel like they are no more required in this world, or they are so severely diseased and they feel like they cannot be treated. Cell also commits suicide. So why a cell commits suicide? Again, the same two reasons. Either the cell is no more required in the body, the function of the cell is already done, its job is already done, or the DNA of the cell is damaged too much, it's beyond repair, so it commits suicide. Cell suicide is known as apoptosis. So in this lecture, I am going to teach you apoptosis. Let's start apoptosis, that is cell suicide. So in simple language, there are two types of cell death, apoptosis and necrosis. What is apoptosis? Apoptosis is cell suicide. Cell suicide. So if you want to define apoptosis, you can see this diagram. In this diagram, you can see a cell. This is a human cell. In the cell, you can see in the cytoplasm, there are multiple enzymes. The name of the enzyme is caspase. Normally, these caspase are inactive. They are not active. But whenever this cell gets the signal can come from the outside, that is extrinsic signal, or the signal can come from inside the nucleus, that is from DNA, intrinsic signal. In both cases, whenever the signal is received by this cell will activate the caspase enzyme. There are many caspase 1, 2, 3, 4, all of them will become activated. They activated after getting the signal. So these activated caspase will start degrading the cell, degrading the nucleus of the cell, degrading the cytoplasm of the cell, degrading the cell membrane, the structural components of the cell. So the complete cell will be converted into multiple apoptotic bodies, you can see in the diagram. In each apoptotic body, there is a small amount, there is a small fragment of the plasma membrane, there is a small amount of the cytoplasm, and there is a small amount of nucleus. Nucleus is fragmented into multiple pieces, the entire cytoplasm is degraded, and plasma membrane is also degraded. So multiple apoptotic bodies are formed. But the point to remember here, to consider here, is that while this process, there is no leakage outside. There is no cytoplasmic leakage outside. That's why there is no inflammation. In the end, a phagocyte will come. Can you see a phagocyte in this cell? In this diagram, this is a phagocyte. In the end, a phagocyte will come, and that phagocyte will engulf all the apoptotic bodies. This is how this cell disappears from the world and the cell is committing suicide. This is apoptosis. So what is the definition of apoptosis? Apoptosis is a pathway of cell death that is induced by a tightly regulated intracellular program. In this pathway, the cell which is destined to die, activate an enzyme known as caspase inside the cytoplasm of the cell, and the caspase will degrade the cell's own nucleus and cytoplasmic component. The cell will be converted into apoptotic bodies, and in the end, a phagocyte will come and engulf all the apoptotic bodies. In the entire process, there is no leakage, there is no inflammation. Why I am highlighting these two terms, there is no leakage and no inflammation? Since there is no leakage, there is no inflammation. In the entire process, there is the plasma membrane remain intact. Plasma membrane get converted into multiple apoptotic bodies, but leakage is not there. This is the feature which differentiate apoptosis from necrosis, the other type of cell death. There are two types of cell death. I have told you in the beginning only, apoptosis and necrosis. So in apoptosis, there is no leakage, no inflammation, because the plasma membrane is intact till end. But in necrosis, the cell membrane, the plasma membrane disrupts, because of which leakage of cytoplasm is present and because of which inflammation is present. These are the hallmark differences between apoptosis and necrosis. So this is the definition of apoptosis that I have told you. Now let me start the mechanism. Let me teach you the mechanism of apoptosis. As I have told you, when the cell is destined to die, that cell will receive the signal. The signal can come from the outside in the form of the ligand, and the signal can come from inside the nucleus. When this cell is no more required, its job is done, its function is done, the signal will come from outside in the form of the ligand, and that pathway will be known as extrinsic pathway. Extrinsic pathway means signal will come from outside. And when the signal will come from the damaged nucleus. The nucleus, the DNA is damaged beyond repair. It cannot be repaired anymore. The damage is too much. The DNA will give signal to the cell that I am damaged, please commit suicide. So such signal is coming from inside of the cell. It is known as intrinsic pathway. So in the mechanism, I am going to teach you two pathways, extrinsic and intrinsic. In each pathway, extrinsic also and intrinsic also, there are two phases, initiation phase and execution phase. So this is the thing in the flow chart. You can see there are two pathways, extrinsic pathway, intrinsic pathway. In each of the pathway, there are two phases, initiation phase, execution phase. Now please understand, I will teach you the initiation phase of execution pathway first. Then I will teach you initiation phase of intrinsic pathway, right? And in the end, I will teach you execution phase of both of them together, because execution phase of both of them is convergence point and it is common for them. So I will teach the execution in common. So my sequence in this lecture will be first initiation of extrinsic pathway, then initiation of intrinsic pathway, and in the end execution of both pathways together. So listen it like a story. Let me start initiation phase of extrinsic pathway. We are starting with extrinsic pathway and with the initiation phase. So this is the extrinsic pathway initiation phase of extrinsic pathway. So in this, you can see it is on the surface of all cells, there are certain receptors. You can see a cell in this diagram. On the surface of all cells, there are certain receptors which are known as death receptors. Death receptors are of two types. What are the two types of death receptors? The first is known as FAS, CD95, FAS or CD95. The second is TNF, tumor necrotic factor. These are the two types of death receptors present on the surface, but they are inactive. They are present on the surface of all cells, they are inactive. Now see, here is a sketch diagram in front of you. Can you see a cell in this diagram? Can you see the nucleus of the cell? Inside the nucleus of the cell, there is double-stranded DNA. It is a very good diagram depicting the information. On the surface of the cell, you can see the red colored receptors which are death receptors. These death receptors are CD95 or FAS, one and the same thing, FAS or CD95 receptor, right? Now, when the cell will get a signal, the signal is coming in the form of the ligand. Can you appreciate a ligand here? The ligand is FAS ligand, that is CD95 ligand. When this ligand is coming here and binding, this ligand is coming. So let me zoom out the portion. I'm zooming out this portion, right? So here in this sketch diagram, you can see the death receptors and you can see here the FAS ligand. When ligand is coming and binding with the death receptor, that is CD95 receptor. So FAS ligand is coming and binding with the FAS receptor. CD95 ligand is coming and binding with the CD5 receptor. So can you see a distance between them? Normally, the death receptors are present at a distance from each other. But whenever ligand is coming and binding with the death receptor, multiple, you can see here the combination. The FAS ligand binding with the FAS receptor. Death ligand binding with the death receptor. Multiple death receptors come together. Appreciate, they all are coming together. And after coming together, they form a domain. Appreciate, this is the ligand. When it is binding with the death receptor, multiple death receptors are coming closer and together and they are forming a domain. What is the name of this domain? It is known as FADD, FAS associated death domain. It is known as FAS associated death domain, FADD. This domain is formed in the cytoplasm, right? Now in the cytoplasm, we have various caspase. As I have told you, all these caspase are inactive. But because of formation of FADD, the first caspase which become activated is caspase 8. Pro-caspase 8 convert into active caspase 8 because of the formation of FADD. So caspase 8 is the first caspase which is activated in extrinsic pathway, initiation phase of extrinsic pathway, right? Point to be considered here. Now the same here, you can see this diagram showing the same information. So here also you can see this is FAS ligand. It is coming and binding with the FAS receptor. When the combination takes place, multiple FAS receptors are coming closer. When multiple FAS receptors are coming closer in the cytoplasm, a domain is formed. The name of this domain is FADD. Can you appreciate this is FADD? FAS associated death domain. Because of the formation of this domain, pro-caspase 8 converts into active caspase 8, right? So this is the story. You can write down in the form of the flowchart the same story. FAS ligand coming to FAS receptor, multiple FAS receptor coming together, forming FADD, and FADD activates caspase 8. So the same story, right? Instead of FAS receptor, the second type of death receptor is TNF receptor. So TNF receptor, for the same story is there. For TNF receptor, TNF ligand will come. So here in this diagram, you can see this is the TNF ligand coming and binding with the TNF receptor. So multiple TNF receptors are coming together. Again, they form a domain. This time, the name of the domain is TRADD, TRADD. So it is TNF receptor associated dead domain. In that, FADD was formed. Here TRADD is formed. Fast associated dead domain, TNF receptor associated dead domain. Ultimately, the ultimate result is same. The end result is same. So here also pro-caspase 8 get converted into active caspase 8. So here also the active caspase 8 is formed. The summary is that for extrinsic pathway in initiation phase, caspase 8 is the first caspase which will get activated. You can see here I am ending the story at caspase 8. So you should be thinking, you may be thinking that what's ahead, ma'am? What will happen ahead? So I will continue the story here in execution phase later on. Let me first tell you the initiation phase of intrinsic pathway. So let me first, then in the execution, I will teach you the two execution phase together. So here I am teaching you initiation phase of intrinsic pathway, right? So here it is intrinsic pathway. Here the signal will come from inside. As I've told you, in extrinsic pathway, the signal come from outside. That's why known as extrinsic. Here in intrinsic pathway, the signal come from inside. That's why known as intrinsic, right? So who will give the signal? C. So can you see a cell in this diagram? Inside the cell, can you see a DNA? The nucleus containing double-stranded DNA. Can you see a mitochondria? This is mitochondria. These are the two membranes of the mitochondria, the inner membrane and the outer membrane. Between two membranes, this is protein known as cytochrome C. So cytochrome C is the protein present between the two membranes of the mitochondria. It is always present between two membranes of mitochondria. It will never come out in the cytoplasm. If it leaks out in the cytoplasm, it will lead to apoptosis, right? So till now, cytochrome C is present inside only. It is present between the two membranes of the mitochondria. Now see this diagram, the zoom version of the same, right? Now mitochondria have permeability. You can say this is the gate. This is the opening gate or the permeability, the gate, the door, the permeability in the mitochondria. So why cytochrome C is not coming out of this permeability, leaking out or coming out of this door? Why? Why? So there are two guards. Understand, there are two guards. The name of the two guards are two proteins which close the door, which inhibit the permeability and prevent cytochrome C to come out normally. The name of the two proteins is BCLX and BCL2. These are preventing apoptosis because they are closing the permeability, inhibiting the permeability and inhibiting cytochrome C to leak out. Once cytochrome C comes in the cytoplasm, it will cause apoptosis. But they are inhibiting cytochrome C to come out. That's why they are inhibiting apoptosis. That's why these two are known as anti-apoptotic proteins. These are anti-apoptotic proteins, right? Right. So till now, this it is very clear. Now imagine a situation that DNA got damaged. Can you see here? The DNA is damaged. P53 is a DNA repair gene. So P53 is activated. P53 tried to repair the DNA, but the DNA is beyond, the damage is beyond repair. It is extensive and it cannot be repaired. So DNA will give the signal to the mitochondria that I am damaged, commit suicide. Commit suicide of the cell. This cell is no more required in the body since the nucleus is damaged, DNA is damaged. So DNA is giving the signal to the mitochondria that start the process of doing suicide, start the process of apoptosis. Here mitochondria is having the central role. It is intrinsic pathway. You can see the signal is coming from inside of the cell. That is from the signal is going from the nucleus to the mitochondria inside the cell only. That's why known as intrinsic pathway. On receiving the signal, what will happen? On receiving the signal in the mitochondria guards will be changed, right? Instead of BCL2 and BCLX, which were inhibiting the permeability and closing the door, the three another guards will come. The name of the guards is BAX, BAD, BIM, BAC. All these BAX, BID, BIM, BAK, BAX, BAD, BIM, BAC. These guards will come and they will increase the permeability. They will open the door, right? So cytochrome C will come out in the next step. So these are known as apoptotic proteins because they are initiating the process of apoptosis. They are increasing the permeability of mitochondria and allowing cytochrome C to come out. So can I say on receiving the signal, the mitochondria replace anti-apoptotic proteins by pro-apoptotic proteins. So the anti-apoptotic proteins were inhibiting the permeability, inhibiting cytochrome C to come out. The pro-apoptotic proteins are increasing the permeability and allowing cytochrome C to come out. So in the next step, you can see this is cytochrome C. It is leaking out. It is coming out in the cytoplasm. It is leaking out. It is coming out in the cytoplasm. After coming here, leaking out and coming out in the cytoplasm, what the cytochrome C will do? How it will cause apoptosis? So again, in the cytoplasm, we are having various caspases. All of them are inactive. So on arriving in the cytoplasm, what cytochrome C will do? See in this diagram. One of the caspases is procaspase 9. It is inactive. It is inactive. How procaspase 9 is inactive? How procaspase 9 is inactive? procaspase 9 is always inactive because it is in combination with APOF1. Because it is in combination with APOF1. So, in the cytoplasm, various caspase are present. All of them are inactive. One of the caspase is caspase 9. It is also inactive. It is in the form of procaspase 9. Who is keeping this procaspase 9 inactive? So, there is a protein known as APOF1. The full form of APOF1 is apoptotic activating factor, APAF, apoptotic activating factor 1. It is keeping the procaspase 9 as inactive. So, it is a rule that till APOF1 is in combination, it is in close contact with procaspase 9 cannot be activated. So, cytochrome C on arriving in the cytoplasm, after leaking from the mitochondria, coming in the cytoplasm, this cytochrome C will take APOF1 with themselves. So, cytochrome C is breaking the pair of APOF1 and procaspase 9. So, procaspase 9 on relieving from APOF1, it will get converted into active caspase 9. So, ultimately, caspase 9 is the first caspase. It is activated in intrinsic pathway. Caspase 8 was the first caspase which was activated in extrinsic pathway. Caspase 9 is the first caspase which is activated in intrinsic pathway. So, right. So, the same you can see in this diagram also. You can see the two layers of the mitochondria. This is inner, this is outer, and between the two layers you can see this is a protein cytochrome C. It is present between the two membranes. This is the permeability. This is the door. Normally, BCL2 is closing the door. BCL2 and BCLX, these are anti-apoptotic proteins. When they receive the signal from the DNA that nuclear damage, DNA damage is there and it is beyond repair. So, they will replace BCL2, BCLX with pro-apoptotic proteins and they open the door. They will increase the permeability. So, cytochrome C leak out. After leaking out, coming in the cytoplasm, it is taking APOF1 with him. So, procaspase 9 will get converted into active caspase 9. So, the same thing is written in the form of the flow chart. You can read the flow chart. You always write flow charts on your exam, right? So, stimulus. What is the stimulus here? Stimulus is DNA damage beyond repair. So, on receiving the stimulus, the mitochondria, these are anti-apoptotic molecules, BCL2 and BCLX, these are lost. And pro-apoptotic molecules like BAX, BAT, BIM, they are coming. So, can I say the anti-apoptotic are replaced by pro-apoptotic on receiving the signal? Yes, right. So, the pro-apoptotic on arriving here, they will increase the mitochondrial permeability, they will open the door. So, cytochrome C will leak out. After coming in the cytoplasm, it activates APOF1. So, it will convert procaspase 9 to active caspase 9. Ultimately, active caspase 9 is formed. So, in this process, the anti-apoptotic proteins, important one are BCL2, BCLX, MCL1 and FLIP. The pro-apoptotic proteins, important one are BAX, BIM, BAT, BIC, P53, APOF1 and cytochrome C. So, anti-apoptotic and pro-apoptotic, two type of molecules are there. So, till now, we have learned that in extrinsic pathway, in initiation phase, caspase 8 is the first caspase which is activated. In intrinsic pathway, in initiation phase, caspase 9 is the first caspase which is activated. Now, what's the story ahead? So, let me start the execution phase of both the pathways together. It is a convergence point. So, here I am starting the execution phase. It is a convergence point for both intrinsic and extrinsic pathway, right? In the execution phase here, you can see in extrinsic pathway, the caspase 8 which is activated. In intrinsic pathway, the caspase 9 which is activated. Now, they are activating the next two caspase, caspase 3 and 7. So, caspase 3 and 7 are the first caspase which are activated in execution pathway. You got my point? And sequentially, all other caspase are also activated. One by one, they are also activated. After activating all the caspase are activated. You got my point? So, here, if it is extrinsic pathway, caspase 8 is the first caspase which is activated. If it is intrinsic pathway, caspase 9 is the first caspase which is activated. And ultimately, they are activating all other caspase. So, all caspase, after activating, what they will do? They will degrade the nucleus, cytoplasm and cell membrane of the cell. So, that entire cell will be converted into multiple small apoptotic bodies. In the end, a phagocyte will come and you can see the phagocyte will engulf all the apoptotic bodies. So, this is the process of apoptosis. So, initiation phase caspase are caspase 8 and 9. Execution phase caspase are caspase 3 and 7. So, these are the caspases that we have understood. In the end, there is phagocytosis. So, whether it is extrinsic pathway or whether it is intrinsic pathway, the last process is phagocytosis. Now, you can see this diagram. You can see here a phagocyte is coming and engulfing all apoptotic bodies. Now, in this diagram, can you think this is a phagocyte? Please appreciate this is a phagocyte, this one, and this is a normal cell. This one, this cell is a normal cell and this cell is apoptotic cell. Here apoptotic bodies are formed. It is apoptotic cell. Apoptotic bodies are formed. Now phagocyte have to engulf this cell but not a normal cell. How does phagocyte recognize the apoptotic cell? How does phagocyte recognize it is a normal cell and phagocyte don't engulf normal cell and it is an apoptotic cell? Phagocyte have to engulf apoptotic cell. There may be some tagging, there may be some marker. So what is that marker? The name of that marker, the name of that tagging is phosphatidylserine. So in this diagram, you can see this cell is a normal cell. This cell is a normal cell and this cell is a apoptotic cell forming apoptotic bodies, apoptotic cell. Now compare. This is the cell surface of a normal cell. On the cell surface of a normal cell, you can see a protein which is present on the inner side of the cell membrane. The name of the protein is phosphatidylserine. It is written as PS here. Phosphatidylserine is the protein. Phosphatidylserine is the protein. In apoptotic cell, so the flip-flap takes place. That is, in flip-flap, externalization of phosphatidylserine is taking place. You can appreciate the cell membrane in apoptotic cell, but this while forming the apoptotic bodies, this phosphatidylserine externalization take place and it is coming on the external surface. This phosphatidylserine is acting as a tagging for the phagocyte. So phagocyte will engulf those cells which is having phosphatidylserine on the outer surface of the cell membrane, not on the inner surface. So who is causing the tagging? The tagging is caused by phosphatidylserine. So in this flowchart, you can see it is externalization of phosphatidylserine which is causing the tagging due to the flipping of inner membrane to outer membrane during the formation of apoptotic bodies. And a phagocyte will engulf only those cells which have phosphatidylserine on the outer surface. That's it. So this is the process of phagocytosis. In this diagram, the same thing is depicted. See, this is the cell membrane. On the inner surface of the cell membrane, this is phosphatidylserine. It is a healthy cell. But again, see the next cell, it is an apoptotic cell. Appreciate it is an apoptotic cell, and on apoptotic cell, the externalization, the flipping already take place, and you can see the phosphatidylserine is on the outer surface, outer membrane. So this is the thing. Now, the next is morphological changes in apoptosis. How many morphological changes? Can you identify an apoptotic cell? What are the features of apoptotic cell? There are 7 morphological features which I am enumerating one by one in the form of this diagram. So this is a healthy cell. When this healthy cell, when this normal cell undergoes apoptosis, the earliest feature is cell shrinkage. Can you appreciate the size of the cell is reduced? So this is known as cell shrinkage. The size of the cell is reduced. So the first change is cell shrinkage. It is always cell shrinkage. It is reducing in size. Number 1, right? That is the first change, earliest change. Very important question for university exams. The earliest change in apoptosis is cell shrinkage. In contrast to necrosis. In necrosis, the change will be cell swelling. Cell swells in necrosis, but cells shrink in apoptosis, right? In contrast. You can see in this diagram, can you see the cells marked by the arrow? This one and this one. These cells are smaller in size as compared to the surrounding cells. This cell is smaller in size as compared to surrounding cell. Come on the next point. The next point is eosinophilia. The next point is intense eosinophilia. As I have told you, the cell is shrinking in size. Because of the cell shrinkage, the cell organelles which are present in the cytoplasm, it can be mitochondria, it can be endoplasmic reticulum, it can be ribosomes, they are coming closer. They are becoming compact. Because of closeness, compactness, the pink color of the cytoplasm is more intense. That is known as eosinophilia. So the second feature is eosinophilia. First is cell shrinkage, second is eosinophilia. So you can appreciate the same diagram here. These two cells are more pink as compared to others. And this cell is more pink as compared to other. So these cells are smaller also, cell shrinkage also, and more pink as compared to other. Coming on the third feature, here also the cell marked by the arrow, it is smaller as compared to other, cell shrinkage is there, and it is more pink. The cytoplasm is more eosinophilic as compared to other. Coming on the third feature, look at the nucleus. The nucleus will become compact. The first nucleus will become compact, that is pycnosis, and after that, fragmentation of the nucleus take place. Fragmentation means nucleus will fragmented into multiple pieces. So you can see after that the nucleus, appreciate the nucleus here and appreciate it is becoming more compact here. So the next change is pycnosis and after that it is fragmenting into multiple pieces. So you can say it is karyorrhexis. So the first was cell shrinkage. Let me write here cell shrinkage. After cell shrinkage, it is seen in this diagram. After cell shrinkage, the second one is eosinophilia. It is also seen in this diagram. So the first is cell shrinkage, then eosinophilia, then third is pycnosis, fourth is karyorrhexis. So you can see the sequence. The next pycnosis karyorrhexis is also appreciated in this diagram. See, pycnosis is condensation of the nucleus. The nucleus is condensed, and carrier access is splitting, fragmentation of the nucleus. Appreciate here in these cells. Can you appreciate a cell marked by arrow? Look at the nucleus. The nucleus is pycnotic here. It is condensed. The nucleus is pycnotic here. In this diagram, these are electron microscopy diagram. These are not light microscopy. This is electron microscopy. In the first, you can see the pycnotic nucleus. This is a cell. Appreciate the condensation. And in the second cell, appreciate the fragmentation of the nucleus. The nucleus is fragmenting. So this is a pycnotic cell. This is a karyoraxis cell. So you can appreciate the changes in the nucleus. The fourth change is on the cell surface. On the surface, multiple blabs are formed. Can you appreciate here? Blabs. Appreciate the blabs here. These are the blabs. So multiple blabs are formed on the surface. The next change. But still, blabs are formed. But the plasma membrane is intact. It is not there are defects in the plasma membrane. No, the plasma membrane is still intact. The next change, can you see? Yeah, in this electron microscopy also, this is a normal cell and this is the apoptotic cell. On the apoptotic cell, appreciate the blabs. Please appreciate these blabs. So in the next feature is formation of apoptotic bodies. Now the entire cell will convert into multiple small apoptotic bodies. Can you appreciate? And in the end, a phagocyte will come and engulf all the apoptotic bodies. So these are all the changes depicted in front of you. Can you enumerate them by sequence? There are 7 changes. The first one is cell shrinkage. It is seen in this diagram. The first one, shrinkage. The second is eosinophilia. So these two are seen in this diagram. The shrinkage is there, eosinophilia is there. Then nucleus is pycnotic. Then nucleus is showing carrier access. Then blabs are there. Then apoptotic bodies are formed. And in the end, phagocyte will come and engulf them. So that is phagocytosis. So these are the changes in sequence. So the earliest one, if I ask you the earliest one, answer is shrinkage. If I ask you most specific one, so specific one is the nuclear changes, that is pycnosis and carrier access. So 2 different questions, 2 different answers. Which is the most common, which is the earliest morphological feature in apoptosis? Answer is cell shrinkage. Which is the most specific feature, morphological feature of apoptosis? Answer is nuclear changes, that is pycnosis and carrier access. So that is the thing. Now, we have completed apoptosis mechanism and morphological feature. Let me tell you a few differences between apoptosis and necrosis, the 2 types of the cell death. What are the differences between them? Apoptosis takes place of single cell. Necrosis takes place of a group of cell, contiguous cell, never single cell. In apoptosis, the cell shrinkage takes place. In necrosis, cell swelling takes place. In apoptosis and karyosis, the nuclear features are common. That is pycnosis, carrier access, the nuclear features are common. In apoptosis, now listen, in apoptosis, the cell membrane is intact. Since the cell membrane is intact, there is no leakage, no inflammation. But in necrosis, on the contrary, the cell membrane is not intact, it is disrupted. So holes are there, gaps are there. So there is leakage. The cytoplasm leak out, and because of leakage, there is inflammation. So please learn the differences. According to the cell, apoptosis occurs in single cell, necrosis occurs in multiple cell. According to the size, in apoptosis, there is cell shrinkage. In necrosis, there is cell swelling. Right? Nuclear changes are same. And the cell membrane. In apoptosis, cell membrane is intact. In necrosis, the cell membrane is disrupted. Holes are there, gaps are there. So in apoptosis, no leakage, no inflammation. In necrosis, there is leakage, there is inflammation. So these are the hallmark features between 2 types of cell death. You can see the same features here, the 2 type of differences here also. In these 2 diagrams also you can appreciate apoptosis and necrosis. So see this cell is showing apoptosis, this one. This one is apoptosis. This side is apoptosis. This side is necrosis. You can appreciate this is apoptosis and this is necrosis. Right? So the normal cell undergoing cell shrinkage, appreciate the size of this cell and appreciate the size of this cell. Here cell shrinkage is there. Here there is shrinkage. And here there is swelling. Here there is swelling. Here plasma membrane is intact till end and apoptotic bodies are formed and phagocyte is engulfing it. So no leakage, no inflammation. On the contrary, on the necrosis, you can see disruption in the cell membrane. So cytoplasm is leaking out, causing inflammation. You can appreciate the neutrophils here. Neutrophils means inflammation. So inflammation always present in necrosis, inflammation always absent in apoptosis. So that is the difference. So what we have learned in this lecture, what is a summary? What we have learned in this lecture. So in this lecture, we have understood apoptosis and necrosis are the 2 types of cell death. Among them, apoptosis is cell suicide. Cell will commit suicide if it is no more required in the body, its job is done, its function is done, or the DNA is damaged beyond repair. What are the mechanism of apoptosis? There are 2 mechanisms, depending from where the signal is coming. If the signal is coming from outside, it is known as extrinsic pathway. If the signal is coming from inside, it is known as intrinsic pathway. Both the pathways have two-phase initiation, execution, initiation, execution. In extrinsic pathway, caspase 8 is the first caspase which become activated. In intrinsic pathway, the caspase 9 is the first caspase which become activated. So what is the mechanism of extrinsic pathway? Signal comes from outside. So signal come in the form of the ligand, right? Either fast ligand or TNF ligand. So multiple death receptors will come closer. They will form a domain, either FADD, fast associated death domain, or TRADD, TNF receptor associated death domain, and they convert procaspase 8 into active caspase 8. So caspase 8 is the caspase which is activated in extrinsic pathway. What about intrinsic pathway? In intrinsic pathway, if the nucleus is damaged, it will give a signal to the mitochondria. So here the signal is received by internal. It is the nucleus giving the signal to the mitochondria. So it is intrinsic. It is inside the cell. So on receiving signal, the mitochondria replace the anti-apoptotic proteins by pro-apoptotic proteins. BCL2, BCLX by Bach's bath BIM. So anti by pro. So pro-apoptotic proteins, they will open the permeability, they will increase the permeability, they will open the door. So cytochrome C will leak out. Here is the cytochrome C, it will leak out, it will come in the cytoplasm, and after coming in the cytoplasm, this cytochrome C activate caspase 9. So caspase 9 is the first caspase which get activated here in this intrinsic pathway. And ultimately, all other caspases are activated in execution pathway, and they will degrade the cell, convert the cell into apoptotic bodies, and phagocyte will come and engulf it. How phagocytosis takes place? Who is doing the tagging? So as I've told you, this is normal cell. On normal cell, phosphatidylserine present on inner surface of the cell membrane. But in apoptotic cell, the phosphatidylserine is flipped on the outer surface. So phosphatidylserine is the protein. The name of the protein is phosphatidylserine, which is causing tagging. A phagocyte will engulf only those cells which is having phosphatidylserine on the outer surface, not on internal surface. So this is the phagocytosis, how phagocytosis takes place. Last thing we have learned in this lecture is the morphological changes in apoptosis. So there are 7 morphological changes, all are visible in this diagram. The first is cell shrinkage. Cell is decreasing in size, and due to decrease in size, there is compactness and it increases eosinophilia. The third is pycnosis of the nucleus. The fourth is karyotaxis of the nucleus. Then blasts are formed, multiple on the surface, but cell membrane is intact. After that, apoptotic bodies are formed, multiple, and in the end, a phagocyte will come and engulf all apoptotic bodies. That's it. We have learned the differences between apoptosis and necrosis, the two types of the cell death. Apoptosis takes place of single cell, necrosis multiple cell. Apoptosis, cell shrinkage, necrosis, cell swelling. In apoptosis, cell membrane is intact. In necrosis, cell membrane is disrupted. Since in apoptosis, cell membrane is intact, so there is no leakage, no inflammation. In necrosis, on the contrary, there is leakage, there is inflammation. So these are the hallmark differences between apoptosis and necrosis. So that's all about it.